Hi, yeah. Have a seat anywhere. Welcome to the Tutoring Center. Here, I'm Ray. Nice to meet you. Yeah, yes, I saw you there too. Well, it's good to finally actually meet. So today, I saw on the form you put biology. Okay. Oh, okay. So you have a test on Tuesday. Okay. So yeah, I prepared all of the notes I need for that class. So let me just go ahead and pull everything up and let you know kind of what we're doing. But yeah, we have just a little bit of time allotted right now. So yeah, isn't this a great resource to be able to use to get free tutoring whenever you need? So I'm really glad you came in. So what I think we should do is take a practice test. And then we can kind of go over the practice test. Okay, cool. So, okay, so the first four are multiple choice. Okay. So number one, Charles Lyle championed the idea of, so A is the law of biotic potential, B is the Hardy-Weinberg equilibrium, C is taxonomic classification, D is the law of segregation, E is uniformitarianism. Okay. Number two, when selection favors the middle range of variation within a trait, for example, average birth rate rather than very small or very large, this is called A, balancing selection, B, stabilizing selection, C, directional selection, D, founder's effect, or E, none of the above. Okay. 3. A trait, like hair or eye color, that is coded for by more than one locus is called it's a specific point on a chromosome. Yeah. Yeah, like a gene. So A, polygenic, B, plyotropic, C, Mendelian, D, polymorphic, or E, simple. Oh, you changed your mind? Yeah, I'll read them again. Okay. A, polygenic, B, plyotropic, C, Mendelian, D, polymorphic, or E, simple. Okay. Yeah. 4. The Hardy-Weinberg equation is A, the relationship between alleles in a gene pool of similar individuals, B, the frequency of alleles for any given trait, C, a way of determining whether or not a population is in equilibrium, D, the method used by Darwin to determine the adaptation of finches? Yeah, the Hardy-Weinberg equation. Okay, so that's it for the multiple choice. So then there's 2 short answer here, but let's save those to the end so we can like just discuss them. Okay, yeah, so there's actually 4 short answer. So then let's just do the last one. Okay, so for 9 through 13, you can either answer mitosis, meiosis, or both mitosis and meiosis. So for example, the first one happens in the body cells. And so that one would be just mitosis. Right, okay. So then the next one is produces 4 daughter cells. Would that be mitosis, meiosis, or both? Okay. Begins with 46 chromosomes. Produces 2 daughter cells. And has 1 nuclear division. Okay. So let's check your answers. Do you think this is helpful? Yeah. Okay, so for number 1, Charles Lyle championed the idea of, you said D, the law of segregation. So actually that would be attributed to Mendel. So he came up with the law of independent assortment and a law of segregation. That has to do with how genetic traits are inherited from our parents. Charles Lyle was he was a geologist. That's the word I was looking for. And he basically found evidence that the Earth was much older than it was believed to be at the time. So a lot of people believed that the Earth was only 5,000 years old. And he studied silt deposited by the Mississippi River in the Mississippi, the Delta. So, and then by studying that, he found that the Earth was actually much, much older than we had previously thought. And so he championed the idea of uniformitarianism, the idea that changes in the Earth didn't happen with big changes like Noah's Ark, the flood, that sort of thing. That would be called catastrophism. Uniformitarianism basically says the Earth changed over time due to small, regular changes. Yeah, so that's what Charles Lyle did, geologist. And then you can take this home. I'm just going to make some notes about what we talked about. Mississippi River. Catastrophism. Mendel. Law of assortment. Okay, so then for number 2. When selection favors the middle range of variation within a trait, for example, average birth weight, rather than a small or very large birth weight. This is not my test. This is actually used from a previous year that I was given for tutoring purposes. But anyways, this is called, so you said balancing selection. The answer would actually be stabilizing selection, but this one is a bit confusing. And so let's see. Balancing versus stabilizing. Balancing selection is when when natural selection would favor the heterozygous, basically. So someone who would have both a dominant and a recessive allele. So for an example of this would be sickle cell anemia. Now I'm sure you've probably heard this example, but maybe it could be helped to have it explained. Yeah, and so basically that would be if you have 2 recessive alleles for sickle cell, you are going to have a lot of problems. They're gonna kind of clog up your joints, and you have these, they're like sickle-shaped, little moon-shaped blood cells rather than regularly shaped blood cells. And so that can be quite painful and cause a lot of issues. 2 dominant is pretty normal for anyone else, for most populations. But in populations where malaria is quite common, sickle cells actually are beneficial because the malaria parasite can't complete its cycle with that type of blood cell. And so in areas where malaria is, there's a lot of fur in the air. In areas where malaria is more common, you will find this balancing selection where they won't have all the normal cells because the sickle cells help with the malaria, but they won't have the sickle cells because those are not healthy. And so they'll have a combination they'll have both regular shaped cells and sickle cells. So that's what balancing selection is. And stabilizing selection is what is described in the question. So height, birth weight, anything that basically says it's not going to favor an extreme on one end or the other. It's going to natural selection is going to make it more even. And like, for example, birth weight in this, right? If you have a really large baby, it's not going to be born easily. The labor would be too difficult. And if you have too small of a baby, well, obviously, very small infants or premature babies have struggle to survive past their first few weeks. So that's why natural selection would favor. Does that make sense? Yeah. And so I'm just going to kind of write those examples. Stabilizing Okay. For number 3, a trait like hair or eye color that is coded for by more than one locus. Go ahead and it's called, so you said Plyotropic, and the answer was actually Polygenic. These are another 2 that get easily mixed up, so I could totally see why you might have said that. So plyotropy is basically when one gene codes for multiple different traits. So an example of this would be a gene that we have in people that codes for both earwax and body odor. Yeah, it's really interesting. So those who have a type of like gray, flaky earwax actually have less body odor than those who have like yellow, sticky earwax. So it's super interesting. But that would be an example of plyotropy. Polygenic is the reverse of that. When one trait, like as it says in the question, hair or eye color, is coded for by multiple genes. So you can kind of see that in the word polygenic, many genes. So I'm also going to make that a note on here. Polygenic. and just marking the right answers as well. And then okay, and then the last multiple choice. 4. The Hardy-Weinberg equation is, this was kind of a tricky question, but you did get it right, a way of determining whether or not a population is in equilibrium. So some of the other questions on the back actually do cover the Hardy-Weinberg equation, and this is kind of a bigger concept that we want to make sure you understand for the test. But let's just quickly go back to the last question, and you actually got these all right, so. But yes, mitosis. Yeah, meiosis and mitosis you've kind of covered a lot. It does seem a lot of genetics, biology, some of this stuff about DNA replication, cell replication, cell cycle. All those things might have been covered in a previous class, so I kind of see a wide variety of people coming in with little to no knowledge about it to a lot, just depending on how many times they've taken biology and paid attention. So. But yeah, I'll just go over it. Mitosis happens in the body cells. Meiosis produces 4 daughter cells. Both mitosis and meiosis begin with 46 chromosomes. Mitosis produces 2 daughter cells, and mitosis has 1 nuclear division. Yeah. So great work on that. Let's go over the short answer. So number 5 is, what was right about Lamarck's ideas about evolution and what was not right? Yes, yeah, that one is Lamarck. Yeah, no, that's definitely the easier part. So what wasn't right was that he believed that evolution happened on an individual level. He came up with the idea of the inheritance of acquired characteristics. Inheritance. And basically, what that was is that, think of a giraffe. If a giraffe has a very short neck and it needs to reach the leaves on the top of the tree, it's going to stretch its neck just a little bit, stretch it a little more, until it has a longer neck, and then it's going to eat the leaves. And we obviously know that that's not how that works. My child would not be born with tattoos. An amputee parent will not, their child would likely have 4 limbs. So that was not right. The important thing to understand is what was right. And what he realized was that need and the environment influenced change. So the giraffe's need for leaves was what was driving the evolutionary change. Giraffes. Okay. The next question is, does that sort of answer your question? I would recommend looking over that. It is important to understand all of the people that influenced Darwin's work, and influenced kind of what we understand about genetics and evolution today. So, yeah. So, number 6. Why is it difficult to apply the biological species concept to species in the past? So this is less to do with like genetics, more to do with speciation. And I'm not sure, obviously I'm not your teacher, I'm just a humble tutor, but I doubt this will be on the test. But to just basically cover it, it's you can define a species by the biological species concept, the idea that if 2 individuals will naturally reproduce and produce viable and fertile offspring. So that means that they will reproduce naturally without human intervention. That means that their child will be viable, meaning it will live, and that their offspring will be fertile, meaning it can have offspring of its own. So why is it difficult to apply this concept to species of the past? Well, how do we know that they reproduce naturally? How do we know that their child lived? So things like that. You can only really do that in sort of a experimental setting. Yeah. But I think this question is really here to make you look into speciation, because that is kind of the biggest concept when we're looking at macroevolution. 2 more questions on the back. If the frequency of the T allele, and this is a capital T, in the population is 60%, what is the frequency of the little t allele? So if the frequency of the dominant allele is 60%, what is the frequency of the recessive allele? Yeah, it would be 0.4. And so this is where we are going to be using that Hardy-Weinberg equilibrium. Basically, P plus Q equals 1. P being the frequency of the dominant allele, and Q being the frequency of the recessive allele. And it just sort of makes sense. There are 2 options. It's a binary setting where we have dominant and recessive. 100% of the gene pool is made up of that. So whatever isn't made up of whatever the dominant allele doesn't make up, the recessive does, and vice versa. So it'd be 40%, 0.4 would be the recessive. So great job. And then the next question also covers the Hardy-Weinberg equilibrium. So that sort of consists of the 2 equations. Do you know what the second one is? If the first one is P plus Q equals 1? Yeah, close. It's actually P squared plus 2PQ plus Q squared equals 1. And so that's basically just if you square the first equation. Yeah. Imagine it like a crossing, like a reproductive setting. You have to have 2 alleles, so you're kind of pulling 2 instead of 1. Does that make sense? Yeah. I can send you some videos that will be super helpful in studying for the test, and one of them kind of describes where we get that equation from. Understanding where an equation comes from is crucial in understanding how to use an equation. So, with your permission and your email, I can send you a few videos. Also one that illustrates DNA replication in the process, and one that shows you the difference between mitosis and meiosis. I just have a couple of those because they're very helpful. Sorry, but the question is, in the equation p squared plus 2pq plus q squared equals 1, what is represented by each of the following? So what does p squared represent? Yeah. So if p is the frequency of the dominant allele, then p squared is the frequency of the dominant trait. What does 2pq represent? So what does q squared represent? Yeah, the recessive trait. So basically those who have 2 recessive alleles. So 2pq then is correct. So if you have both the dominant and recessive, or in other words, if you're heterozygous. Okay, perfect. So you did great. I'm going to go ahead and give you our practice test to look over our answers. And I'm just going to give you a few more materials because we have a few more minutes left. Okay, so I will give you another practice test. And obviously you don't have to do this. I'm not checking or grading. This is all just to help you. So, but if you want another study tool, I'll give you that. And then I have a list of terms that you should study, as well as the review questions to go over in the textbook. Because which textbook are you using? Okay, yeah, so it is the same, and obviously it should be, unless they changed it without telling me, which obviously. So from chapter 1, you need to know, be able to know these terms and concepts. So that would be the subdisciplines of anthropology, scientific method, what a hypothesis, theory, and a law are, natural selection, taxonomic classification, also known as Linnaean classification, the law of biotic potential, inheritance of acquired traits, so that's Lamarck, like we talked about, descent through modification, that's Darwin, evolution, catastrophism, gradualism, uniformitarianism, and adaptation. Those are the terms from chapter 1, and as well as some people to know. That would be Gregor Mendel, Charles Darwin, Carolus Linnaeus, Jean-Baptiste Lamarck, our boy Lamarck, Thomas Malthus, and Charles Lyle. And then the review questions. I would just say to go back into the chapter. There's at the end of the chapter they have a lot of definitions, and so I would just make sure to look at those definitions. I can also send you a link to like a Quizlet. So then the more genetic terms that you should know, DNA, RNA, that includes tRNA and mRNA, DNA replication, protein synthesis, meiosis, mitosis. You need to know what a gene is, a point mutation, a regulatory gene, a homeobox gene, allele, locus, chromosomes, genotype, phenotype, dominant and recessive, codominant, incomplete dominance, heterozygote, homozygote, polygenic, plyotropy, linkage, and crossing over. Yep. Then, yeah, so the videos I'll send you will cover a lot of this stuff. Knowing the difference between these. Yeah, okay. So yeah, on page 98, questions 1, 2, and 4. But it does say that here, so just look at the bottom of the vocab terms. Next, more so population genetics. So this is on a bit of a broader scale than DNA. We're looking at what is a breeding population, what is a gene pool, allele frequencies, gene flow, genetic drift, founder's effect, balancing selection, stabilizing selection, so we went over those, directional selection, disruptive selection, fitness, that would be like environmental fitness, Hardy-Weinberg equilibrium, evolutionary forces, and so those are just some of the above things. Non-random mating / assortative mating, positively assortative mating, and negative assortative mating, yeah. And then sexual selection. So yeah, that just has to do with natural selection, less natural selection, but the other forces of evolution, what drives change in populations and speciation. Yeah, and then the page 136, question 5. Okay, and then lastly, the biological species concept, morphological species concept, lineage species concept. So these are ways of measuring speciation and evolution. Speciation and reproductive isolation. So I'll go ahead and put that with your materials here. Oh, sorry, that's my clipboard. You don't need to take that, but there you go. Do you have any other questions for me? Which one? Oh, yeah, homeobox genes versus regulatory. Yeah, so regulatory genes turn on and off genes in an individual's life. So it's going to regulate growth, making you grow at different rates when you're a baby versus an adult. Obviously, we need some way to regulate growth like that, because if you grew at the rate you did as a child, you would never stop growing. Make for some scary people, you know, you'd be very tall. So yeah, that's what a regulatory gene is. Homeobox genes are very interesting. They're the genes that turn off permanently certain traits. So for example, an example of a homeobox gene would be chickens. Birds are basically dinosaurs. They're quite related genetically. Well, chickens have a gene that codes for teeth, like dinosaurs. The homeobox gene turns that off permanently, so it makes it so chickens don't have those teeth. But some scientists have actually experimented with that gene and isolated it and found a way to turn it off and hatched chicks that had teeth. So that's what a homeobox gene is. Does that answer your question? Perfect. Well, I'll go ahead and email you those videos. Looking through the textbook, looking through your labs. Memory recall. So try not looking at your notes. Try going in cold like a test. So maybe taking this other practice test without notes or anything. Don't be afraid to get things wrong, because it's just you quizzing yourself, just like here. It's just us. There's no grade involved, so yeah. So anything else I can help you with? I know you probably have to get going. Yeah, no problem. And you know what, actually, that's just my phone number. So if you have any questions leading up to the test, feel free to text me. I don't mind at all. So yeah, I'm glad I could help.